ginásio, e também o Dia Mundial da Leucemia crônica, que é no dia 22 de setembro, por conta da convocação no 22, ficou esse dia para celebrar, né? Então, vocês que estão... Como é que eu faço para essa slide aqui agora, hein? Peraí. Então, gente, vocês nasceram numa época, né, que quando vocês nasceram, década de 80, a sobrevida era assim, envelhecer, né? Era bem ruim, a maioria dos pacientes evoluíram com plástica. E hoje em dia, aqui é o progresso ao longo das décadas, né? A gente vê então, que hoje em dia a sobrevida é de 90% 10 anos, então o desenvolvimento dessa classe, né, de medicamentos e dos possibilitou que a gente mudasse a história natural da leucemia. A gente sabe que nem todos os pacientes têm essa evolução, né, e justamente a aula de hoje vai focar bastante nisso, né, nos motivos. Essa aula foi apoiada pela Fundação, né, Internacional de Leucemia Crônica. Aqui eu estou o endereço do site, é uma fonte muito rica, né, de informação médica, de aulas, né, aulas e encontros, né, de grupos de discussão de leucemia crônica. Essa fundação ela apoia os lectorships, a gente tem esses fóruns, né, no Oeste, também tem o congresso anual de LMC também. No site vocês podem ter acesso à discussão de caso clínico, tem várias publicações relacionadas à LMC e o alcance dela é mundial. A gente tem também alguns projetos em comum, né, que a gente participa, relacionados então a uma aliança genômica, a TFR Alliance, que é um grupo que está focando na questão da descontinuação do tratamento, entre outros trabalhos que a Fundação tem feito. Então, a nossa convidada especial de hoje é a doutora Simone Soverina, professora da Universidade, do Departamento Médico e Cirúrgico da Universidade de Bolonha. Ela também é chefe, né, da área de pesquisa de leucemia mieloide, também no Hospital, né, de Bolonha. Ela tem mestrado, doutorado, então, em hematologia experimental pela Universidade de Bolonha, e ela tem um foco muito grande na pesquisa dela sobre leucemia mieloide crônica e mastocitose. Ela publicou já centenas de publicações e ela é bem conhecida como a rainha das mutações, tá? Ela é uma das principais pesquisadoras nessa área. E ela faz parte de diversas sociedades europeias e americanas em hematologia. Então, so Simone, it's a great pleasure to have you here. It's a privilege to hear you talking to us about the mutation. Thank you very much, Simone.
Thank you, Katia. Good afternoon to everyone. So, dear colleagues and friends from Brazil, it's a great honor and a pleasure to join you virtually today to celebrate the 25 years of TKI therapy, sharing some memories of the achievements we have reached so far, and some thoughts about what translational research and advanced diagnostics can still do to support clinicians in further optimization of CML management. So CML, as you know, is a remarkable example of how, from the bench to the bedside and back, biological acquisitions may drive a therapeutic improvement, and how clinical and diagnostic needs may drive translational research and technological advances, respectively. Since the very dawn of the tyrosine kinase inhibitor era, the synergy between the lab and the clinic has been instrumental, as you know, for therapeutic success. And the inextricable link between the lab and the clinic is testified by the fact that molecular response is the gold standard for the evaluation of therapeutic efficacy. The fact that key molecular response levels are treatment milestones and endpoints, the fact that regular monitoring of BCR-ABL transcript levels and kinetics are key triggers for mutation testing and therapeutic reassessment, and the fact that BCR-ABL kinase domain mutation testing is an important guide to TKI selection. Those of you who have witnessed the advent of imatinib 25 years ago will remember for sure that soon after, there is an echo actually. Okay. Let's try. We'll remember for sure that soon after the first clinical trial started, cases of resistance were reported where activation of BCR-ABL kinase activity was observed due to the acquisition of a mutation in the kinase domain impairing imatinib binding, and demonstrating that BCR-ABL was actually not so easy to tame, at least not in all the cases. You will also remember that the first mutation to be described was the T315I that has remained our worst enemy for many, many years until the advent of the third generation TKI, ponatinib. But as more and more studies of imatinib-resistant patients were published, more and more mutations popped up. And here you can see an incomplete overview of all the mutations, just the most frequent mutations that have been reported in association with imatinib resistance, with highlighted in red those retaining insensitivity to second generation TKIs. As you also know, the problem of resistance and mutations has been one of the major engines that drive the development of second-generation TKIs, dasatinib, nilotinib, and bosutinib, of the third-generation TKI, ponatinib, and all of these belong to the so-called family of the orthosteric or ATP competitive inhibitors, those binding to the active site of the kinase. But more recently, another member of a different family, the family of the allosteric inhibitors, has been developed and has entered the clinical use. And I'm referring to asciminib, that is really the forerunner of this novel category of inhibitors, the allosteric inhibitors. And the allosteric TKIs, as you can see here from the cartoon, bind elsewhere, not in the ATP binding site, but rather to sites or surfaces that are critical for the regulation of kinase activity. And usually these sites are very kinase specific in compared to the ATP binding site. As far as ABL and BCR-ABL are concerned, in particular, the development of this novel allosteric inhibitor was made possible by the knowledge of how ABL kinase activity is regulated by myristoylation. As you can see in the slide, the N-terminal cap of untranslocated ABL undergoes a post-translational modification called myristoylation and binding of myristate to a narrow hydrophobic pocket in the C-terminal lobe of the kinase domain physiologically inactivates ABL and also its cognate kinase ABL2. So this is a very specific mechanism of kinase regulation only for ABL1 and ABL2. As you know, as a result of the translocation, the N-terminal part of ABL is lost and is replaced by sequences deriving from BCR. So this physiological control mechanism of kinase activity is lost as well. And so the rationale that led to the development of asciminib was actually to recreate a molecule structurally mimicking myristate in order to be capable to bind to the same hydrophobic pocket in the C-terminal lobe of the kinase and induce the same conformational rearrangement leading to shutting down kinase activity. So both myristate and asciminib binding induce this conformational change you can see here in the slide, stabilizing the closed inactive conformation of the kinase. And since asciminib and molecules like asciminib bind to this ABL myristate pocket, they are also called stamp inhibitors, which stamp meaning specifically targeting the ABL myristate pocket. This specific name given to these inhibitors, stamp inhibitors, reflects the fact that allosteric inhibition of ABL and BCR-ABL can be achieved through different mechanisms. And so the mechanism of action of asciminib is only one of the many ways we may have to allosterically control kinase activity. This was just the first one to be clinically accomplished. Those of you who read the first reports of the preclinical activity of asciminib of ABL001 at that time will remember that at the beginning it was predicted that mutation profiles of asciminib and ATP competitive inhibitors, like for example nilotinib, were anticipated to be non-overlapping, right, because asciminib is known to bind to a very distant and distinct region as compared to ATP binding compounds, ATP competitive compounds. However, you are also aware of the fact that subsequent clinical observation have surprisingly contradicted this prediction. So in this table here, I have summarized all the data we have coming from the preclinical studies as well as from the clinical trials of asciminib in chronic myeloid leukemia patients, starting from the phase 1 trials and coming also to the first-line studies, the first results of the first-line studies of asciminib in CML patients. And you can see all the mutations that have been reported in association with asciminib failure, and you will see that while in light blue I have highlighted those mutations that were expected to occur, that is, those mutations mapping to the myristate binding pocket. There are mutations that were definitely unexpected. For example, those highlighted in red are mutations known to confer resistance to ATP competitive inhibitors, so well-known mutations, but completely unexpectedly linked to asciminib resistance because mapping to regions quite distant from the myristate binding pocket, as you can see here in this cartoon. And also in the phase 1 study, they reported even a couple of mutations outside the kinase domain in the SH3 domain of the molecule. So in order to understand why more mutations than expected have been associated in clinical trials with resistance to asciminib, we need first of all to take into account how BCR-ABL mutations may trigger resistance, and in second instance also what is the exact mechanism of action of asciminib. So, as far as how BCR-ABL mutations may trigger resistance, it is important to bear in mind that the impairment of drug binding, either sterically or electrostatically, is only one of the ways that allow mutations to confer resistance. They may also confer resistance by stabilizing the active state of the kinase, and this is the case of many unexpected mutations conferring resistance to asciminib, but they also may confer resistance by increasing TKI activity and oncogenicity. So impairing drug binding is only one possibility for mutation to act, but is not the only one. And as I said before, if we really understand how asciminib works and how asciminib accomplishes kinase inhibition, it will be much easier to understand why so many mutations outside the myristate binding pocket may be associated with asciminib resistance. In this cartoon, you can see the conformational changes that ABL or BCR-ABL undergo while transitioning from the active to inactive state of the kinase. As you can see on the left-hand side, in the active or open conformation, the only intramolecular interactions we can see are between the SH2 domain and the N-terminal lobe of the kinase domain. Upon asciminib binding, there is a dramatic change of the conformation, really a closure of the structure and a reorganization through which, as you can see, the SH3 domain makes contacts with the N-terminal lobe of the kinase, and the SH2 domain make contacts with the C-terminal lobe of the kinase. And all these molecular interactions that I have highlighted here are very, very important to stabilize the active conformation on one hand or the inactive conformation on the other end. So any perturbation that prevents the adoption of or disassembles this closed conformation will confer resistance regardless of whether asciminib binding is retained or not. So this is the key to understand whether mutations even far outside the myristate binding pocket may be capable to confer resistance to asciminib. If there is a stabilization of the active conformation, then asciminib will not be able to induce the conformational change required to shut off the kinase. So, as you can see from this cartoon, that recaps a little bit what we currently know about mutations conferring resistance to asciminib. The mutational vulnerabilities of this drug extend beyond the myristate binding pocket, so we may have mutations, and we are actually seeing quite frequently mutation within the myristate binding pocket, like the A337T or V, and they indeed work by impairing asciminib binding. Into 44V mutation, that is a mutation in the N lobe of the kinase, usually conferring resistance to imatinib and nothing else among the second generation TKIs or the third generation TKIs. Well, the M244V may confer resistance to asciminib because it prevents the conformational rearrangement we have seen that is necessary for the kinase activity to be turned off. And also there is a third group of mutations exemplified, for example, by the Y115N mutation that is indeed not in the kinase domain but in the SH3 domain. These indeed are the interface between the SH3 and the kinase domain, and they induce a steric clash that impairs the clamping of the SH3 to the N lobe of the SH1 that we have seen before. So, at the present times, these are the main mechanisms by which mutations may confer resistance to asciminib, but we are still at a learning stage, and it will be interesting to understand if there will be more mutations, and also if some of these mutations can be overcome by a dose increase, for example, to 200 milligrams, that is the dose that we need to inhibit the T315I, and it's tolerable as well. So, each TKI, non-excluded, not even asciminib, has mutational Achilles heels. And here in this slide, you can see a recap of all the mutations that may be problematic for one inhibitor or another. And actually, it is important to bear in mind that, as I mentioned at the beginning of my presentation, this may be more difficult to tame than initially thought, and is a tough enemy sometimes to fight, because we need to bear in mind that most of these patients have an enhanced genetic instability, and patients harboring mutations are even more genetically unstable than the others. And biologically speaking, they have a more advanced disease. So morphologically, they are still in chronic phase, but from a biological point of view, they are more like advanced phase patients. And this was nicely demonstrated many, many years ago by a study, Radic and his group, showing that gene expression profiles of chronic phase patients harboring mutations were totally superimposable to the gene expression profiles of patients with blast crisis. Demonstrating that indeed mutated chronic phase and blast crisis biologically speaking are super diseases. So patients with mutations have a more advanced disease and have a greater genetic instability that sometimes may allow BCR-ABL to find an escape even from the most potent inhibitors. Here, for example, you can see the example of ponatinib and of some variants of the T315I mutation that may become problematic also for ponatinib and have been occasionally reported in some Ph-positive ALL patients or blast crisis patients developing resistance to ponatinib. As you can see here, if the cells are genetically unstable enough, the original T315I mutation may acquire a further nucleotide substitution, converting the isoleucine in methionine or leucine, and indeed the T315M or L mutations may confer resistance to ponatinib because the structural changes of the amino acid impair ponatinib binding. So sometimes BCR-ABL can find a way to escape even from ponatinib. And another nasty way to escape from inhibition is the acquisition of compound mutation. This is something that we really need to prevent because compound mutations are have been demonstrated to be highly resistant not only to imatinib, not only to second-generation TKIs, but in some instances even to ponatinib and asciminib. Actually, compound mutations are thought to be the result of wrong TKI sequencing, because if the original mutant clone is not rapidly and fully eradicated, it may acquire a second hit, that is a mutation resistant to the second TKI, giving rise to this highly challenging compound mutant configuration, two mutations on the same BCR-ABL allele. So sequencing on narrow spectrum inhibitors in some patients may give rise to these highly nested mutant clones. So the importance of routine BCR-ABL kinase domain mutation testing in patients with non-optimal response to TKI therapy is nowadays widely acknowledged, because patients harboring mutations, as I mentioned before, have a biologically advanced disease, so they too represent a more critical category of patients who need to be followed more carefully. They have a higher likelihood of developing additional mutations leading to relapse on second-line TKI therapy. And last but not least, as I mentioned before, and as you all know, the type of mutation may be useful for second or subsequent line TKI selection to optimize therapeutic outcomes and prevent compound mutations. And now that we know that some imatinib or nilotinib resistant mutations may also confer resistance to asciminib, we know that mutation testing is important even if we plan to switch to asciminib. And this is a table taken from the very last version of the ELN recommendations, ultimately published a few months ago in Leukemia. As you can see here, we have in this table, we have already acknowledged the existence of asciminib-resistant mutations, so they have been included in the list. I have mentioned the importance of mutation testing, and in this slide you can see an overview of the history of BCR-ABL mutation testing over the years. Many of you will remember that the first attempts at developing routine mutation testing, trying to support physicians in clinical decision making, date back to the very beginning of the 2000s. And in this slide, you can see an overview of the technologies that have now are more widely available for mutation testing, starting from what has been for a long time the gold standard, and in many instances still is the gold standard, Sanger sequencing, then transitioning to its natural evolution, that is next generation sequencing, and even droplet digital PCR for specific mutations like the T315I or second generation TKI resistant mutation, for example, that may be individually checked to help physicians in clinical decision making. Here you can see an overview of the technical differences between these methods. For time constraints, I won't go into the details. Of course, as you know, next generation sequencing and digital PCR are more sensitive than Sanger sequencing, and this is for sure an advantage. What I would like to remind you is that both next generation sequencing and droplet digital PCR, at least the current technologies we have in our laboratories, still require an amplification step in order to be able to detect the mutations. And so at very, very low PCR-able transcript levels, we are still not capable to detect mutations for technical limitations. So the greater sensitivity of NGS and digital PCR does not mean that we can interrogate patients with the very, very low levels of BCR-able transcript. It just means that we can interrogate many, many more BCR-able molecules, so we can identify even very few BCR-able molecules among the total of BCR-able molecules we are checking. There now are many studies published in the literature demonstrating the advantages of next-generation sequencing over conventional Sanger sequencing. Here, I'm just recalling our Italian Next in CML study, where we prospectively compared these two technologies in a consecutive series of CML patients who had actually the indication for BCR-able mutation testing, so with failure or warning response to TKI therapy according to the ELN recommendations we had at those times. As you can see here, NGS could detect more mutations not only in patients who were apparently wild type by Sanger sequencing, but also in patients harboring major mutant clones by Sanger sequencing, so additional low-level mutations. And as you can see in the table at the bottom, the advantage of next-generation sequencing was quite evident also in the setting of warning patients. So patients destined to subsequently fail TKI therapy and already harboring emerging mutant clone could be very nicely identified by the use of next-generation sequencing. We have also developed a strategy of droplet digital PCR for clinically actionable mutations. You know that droplet digital PCR is a mutation-specific method, so it does not give you a full overview of the mutation status of the whole kinase domain. Individual assays have to be designed for each and every mutation of interest, but if we consider the panel of mutations that confer resistance to second-generation TKIs, and if we consider the T315I mutation, so those mutations that we need to check for first when we are considering a therapeutic switch, then you can see that the panel is not so wide, and we have been able to build a series of assays to be used to discriminate between nilotinib, bosutinib, or dasatinib resistant mutations. And considering that the F359V mutations may also confer resistance to asciminib, and consider that we may quickly add an assay for the M244 mutation, a similar approach may be used even if we are planning to switch to asciminib. So digital PCR is very sensitive, but more importantly, it's very quick. So we need to perform a rapid switch, and we are just interested in excluding the presence of these mutations. Digital PCR may be a nice alternative. The past, the present, and the future of mutations in CML. So far, I have discussed the role of mutations in BCR-ABL, but as you know quite well, we should also discuss the role of mutations outside BCR-ABL, because in recent years, wider and wider application of targeted next-generation sequencing, but also whole exome sequencing or whole genome sequencing, has revealed that chronic phase CML patients are not so genetically homogeneous as previously thought, and that mutations in cancer genes other than BCR-ABL may be detected in up to 16 to 20% of newly diagnosed chronic phase CML patients. These genes are the usual myeloid suspects, or the Icaros gene, but by far the most frequent of these mutated genes is ASXL1, as you know, that can be found to be mutated in approximately 7 to 10% of newly diagnosed chronic phase patients. Also, the Australian colleagues have highlighted that some CML patients in chronic phase at diagnosis, approximately a little less than 20% of these patients, may also harbor genomic rearrangements in the form of structural variants represented by aberrant fusions formed at the time of the T922 translocation, involving the rearrangement of genes or sequencing on the translocated chromosomes. And these are the results of a sort of explosions of the 9 and 22 chromosome at the time of the formation of the translocation, and then of a clumsy attempt by the Philadelphia positive cell to put the pieces together again, resulting in these aberrant fusion transcripts. They have been collectively termed Philadelphia-associated rearrangements, and they have been associated with worse responses, at least to imatinib therapy. In this study, again, of the Australians, you can see that the so-called AGAs, so additional genetic abnormalities, a term that they have coined in order to denote not only cancer gene mutations, but also Philadelphia-associated rearrangements, at least in the Tidal II study, was associated with worse outcomes. More recently, you probably have already seen the study a few days ago published in Blood. The Australians have also published a subsequent study in which they investigated the role of AGAs, so Philadelphia-associated rearrangements and cancer gene mutations, also in the setting of patients receiving first-line second-generation TKIs or receiving first-line asciminib. These were the patients enrolled in the Australasian studies with these testing the frontline use of these inhibitors, and unfortunately they have shown that at least ASXL1 mutations retain a negative prognostic impact even when using more potent second-generation TKIs first line, even when using asciminib first line, which are actually not so good news. An important question from the biological point of view is why do these cancer gene mutations or ASXL1 mutations associate with reduced TKI efficacy? Because one hypothesis may be that they just reflect greater genetic instability or maybe greater genetic complexity or mutation load, and so they just mirror a more aggressive or high-risk disease in general. But of course, we may also assume that they do play a genuine pathogenetic role by activating oncogenic processes additional to BCR-ABL. And these two hypotheses are indeed not even mutually exclusive. Recently, there has been a study investigated the role of AXXL1 in particular, and based on the findings of this study, it seems that by AXXL1 mutations, by activating ALOX5, that is a critical regulator of leukemia stem cell survival, may indeed play a genuine pathogenetic role. And these authors were also able to show a possible way to therapeutic interfere with the effect of these mutations. So I'm pretty sure that in the future, in the near future, we will know more about the role of AXXL1 and how we can neutralize it. But in the meantime, we have already quite a lot of novel treatment strategies on the horizon. In the table on the left-hand side, you can see the TKIs that are currently undergoing clinical development. Olverembatinib is quite in advanced clinical development, as you know. The other molecules are in early clinical development, so we will see what happens. But there are several molecules, even belonging to the family of ATP competitive inhibitors, so traditional TKIs, let's say, or also allosteric inhibitor with a mechanism of action similar to that of asciminib that might soon become available within our therapeutic armamentarium. And on the right-hand side, I have listed also some alternative strategies that are currently being explored, at least preclinically, in order to inhibit misc-Abl. Like another category of allosteric inhibitors, the so-called monobodies, interfering between the SH2 and the kinase domain interface, or BCR-ABL dimerization inhibitors. You know that dimerization is the very first step for BCR-ABL kinase activation, or molecules that are quite famous in several contexts, the so-called PROTACs, proteolysis targeting chimeras. And these are particularly interesting because if we consider that the pathogenetic role of BCR-ABL is not limited to its aberrant kinase activity, but BCR-ABL has also an important scaffold role in triggering the neoplastic transformation of leukemic stem cells, a strategy leading to the degradation of BCR-ABL might be even more effective than a strategy just only aimed at inhibiting BCR-ABL kinase activity. And then we have many possibilities to develop combos between different TKIs, between, for example, ATP competitive and allosteric TKIs. Of course, the most promising and interesting one is between asciminib and ponatinib, but also combos with other inhibitors like JAK2 inhibitors or venetoclax. Some of these combos have already been explored, others are still being explored, and I would not exclude that some of them might be an option for us in the future. So there are intensive progresses on the clinical side, there are intensive progresses on the genomic side. So even though at present we are still using clinical score to stratify our patients at diagnosis, in the near future, I think I'm deeply convinced we will be using hybrid scores integrating clinical and genomic features of the patients, and based on the results of these scoring systems, we will be able really to develop a tailored treatment of our patients, not only a targeted treatment of our patients. So the CMR story continues to unfold. There are still unmet needs and open issues. We'll need to fill the gaps in our knowledge about mutations conferring resistance to asciminib. We still need to investigate the clinical actionability of AXXL1 mutations. What is the best treatment for patients positive for these mutations at diagnosis or at relapse? We need to devise how to best place and integrate next generation sequencing and droplet digital PCR For PCR-able and non-PCR-able mutation testing, currently the ELN recommendations have not included yet myeloid panel sequencing among the mandatory tests at diagnosis, just because of the difficult actionability of these mutations, but things may change in the near future. And last but not least, we will still have to explore the efficacy and the safety of novel TKI and non-TKI-based approach, both from a clinical and from a biological point of view. And that was my last slide. I would like to thank you for your attention, and I will be happy to take your questions.
Okay. Thank you, Simona, for your beautiful presentation. I will open the questions for the audience. So I will ask you a question first. I saw in the estimate you told about the experience you had about the XL1 mutation in patients treated with nilotinib. And there are several groups that are studying that. In some groups, there is nilotinib is not different, but in other groups, patients that use nilotinib, they fail more if they have the AXL1 mutation. And also about the association of imatinib to overcome imatinib with asciminib to overcome this mutation. Can you comment about that, about these different trials?
The audio is not very good, so did I understand correctly that you asked me to comment about the association between imatinib and asciminib in overcoming ASXL1 mutations? And what was the first part of the question?
Part is about some difference in, for example, for imatinib, it looks like imatinib it has a worse prognosis with this XL1. But with the second generation TKI, it's not clear. So I'd like to hear your comment about that.
Yeah, actually, I have shown the Australian studies that show very strong associations. But I did not show our own data, although they were produced in a smaller series of patients. Actually, our data contradict this finding because both in patients receiving nilotinib first line and in patients receiving patients receiving imatinib with proactive switch to nilotinib in case of non-optimal response, we could not see any statistical difference in terms of response rates. It has to be said that ASXL1 mutations are not so frequent. It's 10% as a maximum. Usually it's 7-8%. So probably in some discrepancies we can see also with other published studies in which different clinical outcomes were found to be significant. Probably sometimes there is just a size limitation, a limitation of the sample size we are analyzing. And in this regard, I think that once all of us will be able to contribute to the ICMLF Genomics Alliance with our data, and once the pooled data sets will be able to be analyzed, we will have stronger evidences pro or con the prognostic significance of ASXL1 mutation. So I'm very eager to see whether putting together all our studies, all our results, we will have stronger evidence.
And what about the association? What do you think about the association of asciminib with these other TKIs to overcome? What do you think? The experience of the German group.
We do not have any kind of experience. We have the data, however, coming from the clinical trials, also the fascination study that is running by the Germans. The problem, as a biologist, knowing that some mutations overlap between imatinib, nilotinib, and asciminib, I would expect that the combination of imatinib and asciminib or nilotinib and asciminib will not be 100% effective in preventing the emergence of some mutations. We may still see M244V mutations, we may still see F359V mutations. So mutationally speaking, I would not bet on these combinations. I think that all of us is dreaming to see our combination with asciminib and ponatinib will perform. The preclinical data are very nice, also showing that showing efficacy with very low doses of ponatinib, which should a little bit limit the toxicity issue. And actually, as far as I'm aware, a very small clinical trial testing this combination has just started in Spain. So I'm very eager to see the results. It will be in highly resistant patients, and the number of patients expected to be enrolled is not so high. But I mean, besides anecdotal reports or the case reports we have seen in the literature, it will be the first attempt really to test clinically this combination, which I think that, at least biologically speaking, is the most attractive.
Another question is about the use of next generation sequencing and diagnosis. What is your opinion? Are you ready for this? Does this make any difference in the state that we are, in the state of art that we are now? I'd like to hear your opinion about doing NGS diagnosis for everybody, or not.
So far within the ELN panel, we had decided not to recommend it, not to make it mandatory. For several reasons, both on the technical standpoint, because there are so many different commercial panels, because there are several, we need a uniform way to interpret these mutations, and because of these mutations currently are hardly actionable. We do not know yet what is the best way how to treat these patients. So I think that routinely speaking is not necessary, but it would be welcome on the clinical research side. So whenever we have the chance to screen our patients for these mutations at diagnosis, then we should grab this chance and then just keep the results in the drawer, and then afterwards correlate with response and see if we can build our own experience. But practically speaking, I mean, what would you do if you found an AXXL1 mutation in your patient? It would be very empirical anyway.
Yes, I agree with you. Does anybody have any other question? No? Israel Valeuza? No? Okay, so thank you very much, Simone. It was great to have you here, and I hope to see you soon in Estoril.
Thank you very much to all of you. Have a great remaining of the day.
Thank you. Bye.
Goodbye.